Okay, so welcome to this next video in which we are discussing the adaptive immune response. Okay, so we discussed in the previous video how you activate the humoral immune response. Okay, so we've discussed how you get the activation of naive CD4 positive T cells, okay, and how they become T helper 2 cells. And then what we discussed is how antigen will bind to B cell receptors on B cells within the B cell follicle. So remember, all of the B cells within a B cell follicle will have slightly different B cell receptors, okay? And these B cell receptors will bind to different antigens. However, if you're percolating a huge amount of antigen through these B cell follicles, then eventually what will happen is this antigen will just happen to find a B cell which has a complementary B cell receptor. Now, when that antigen binds to this B cell, it will then be internalized. The entire B cell receptor will be internalized with the antigen bound to it. The antigen will then be broken down and fragments of it will be presented on the surface of the B cell on MHC class 2. In addition, what will happen is that the B cell will be exposed to a huge amount of pathogen-associated molecular patterns. So PAMPs will be also within the lymph that is coming from the afferent lymph vessels. And this will be dousing these B cells in PAMPs, okay, which will activate the pattern recognition receptors of these B cells, leading to these B cells then expressing co-stimulatory molecules such as B7.1 /B7.2 and CD40, okay. This B cell will then interact with T helper 2 cells, so it will present this antigen fragment to the T helper 2 cell, which has the complementary T cell receptor, and the CD4 on the surface of this T helper 2 cell will bind to the MHC class 2. In addition, CD28 and CD40 ligand will bind to the co-stimulatory molecules, which are B7.1 and B7.2 in the case of CD28 and CD40 in the case of CD40 ligand. This will lead to the activation of this B cell, and it will start dividing and dividing and dividing and dividing within the B cell follicle to create this germinal center. Okay, now each of these B cells that it produces are not identical to it. Instead, every single one of them will have a slightly different B cell receptor because all of them undergo a process known as somatic hypermutation, where they slightly mutate the antigen binding portion of the B cell receptor. Okay, now that means that we've got a variable colony here. These different B cells will have differing affinities. Well, they'll have B cell receptors which have differing affinities for this antigen that we are trying to launch a humoral immune response against. Okay? Now, you have these cells known as follicular dendritic cells within B cell follicles, which sort of capture antigen so that there's a good supply of this antigen for these B cells within the germinal centers. Okay, but there is a limited supply is the important principle, and only B cells which manage to get their hands on this limited supply of antigen will be induced to survive, basically. Okay, so those which have which have an improved affinity for the antigen, i.e. their B cell receptor, when it underwent this mutation, increased its affinity for the antigens and is therefore a competitive B cell, that B cell will get antigen, whereas the ones which have suffered mutations which have lowered the affinity of their B cell receptor for the antigen, those will not get the antigen. Okay, so those which don't get the antigen will not get T cell help and they will commit suicide, they will undergo apoptosis. Whereas the ones which do get antigen, they'll go through this process again. They'll internalize the B cell receptor with the antigen, they'll process the antigen, they'll take a fragment of it and present it on MHC class 2, and they'll then go and get T cell help. Okay? they will then be induced to divide again and again and again to produce another whole colony of B cells, which are based this time on them, but which have again undergone somatic hypermutation and therefore all vary. And the idea is that you continue this process and you're gradually producing better and better B cells, which have better affinity B cell receptors. Okay, eventually what happens is one of these B cells decides it's had enough and it's going to start dividing and producing instead plasma cells and memory B cells. So we now want to study what plasma cells are going to do. So plasma cells will leave the lymph nodes and they'll go into the blood, and then once they're in the blood what they produce is antibody. Okay? So antibody is a soluble form of this B cell receptor, so it will have the same binding site that the B cell receptor has, but it won't have this bottom portion that attaches it onto the membrane. So this soluble form of the B cell receptor is known as an antibody. Okay, so this is an antibody. Now, what does the antibody do? Because this is the key component of the humoral immune response. This is what it's all been building up to, the production of this antibody. So the antibody is going to help with the immune attack of this pathogen in multiple ways. So let me discuss these. So basically, if we've got our extracellular pathogen here, so let's say this is our extracellular pathogen, so I'll just label it up as a microbe, okay, then it might have this antigen on the surface, okay? Well, actually there's two types of antigen that we could launch a successful immune response against. Either the antigen is on the surface, or the antigen might be a secreted toxin. So this antigen that we've been launching this immune response against, it might be on the surface of the microbe, or it might be a secreted toxin. Okay, so let's say we've got these two examples basically. It could be a secreted toxin that the microbe has been making, which is incredibly dangerous to the body, or it could just be a surface antigen basically. Okay, and remember the abbreviation for antigen is AG, so I'll put surface antigen there. Right, so if it's a secreted toxin, then what will happen is this antibody that we're producing will come and bind to this secreted toxin, and it will neutralize the secreted toxin. Okay, so this is one of the functions of the antibody. It's the neutralization of toxins. Okay, and I should have just discussed with you the structure of an antibody. So antibodies have this kind of characteristic Y shape here. They have these two heavy chains, 1 and then 2, which are linked together by this disulfide bond here. So I'll highlight this in. So in orange here, these are the two heavy chains of the antibody, okay, held together by a disulfide bond. And in purple, these are the two light chains of the antibody, which are held to the heavy chains by, again, disulfide bonds. So these are light chains. And these are, in orange, the heavy chains. Okay, right. So, if you've got a secreted toxin, and that's what you've initiated this immune response against, then the antibody can bind to that secreted toxin and stop it from having its effect, stop it from being toxic to the body, and that's obviously of incredible benefit. Alternatively, if it's just a surface antigen on the surface of the microbe, then what can happen is the antibody can bind here, and it can do two things, okay? So firstly, it can activate the classical complement pathway on the surface of the microbe. So it can lead to the activation of the classical complement pathway. And I have an entire video on the classical complement pathway in which we discuss this in detail. And the classical complement pathway can then lead to the destruction of this microbe. Okay. In addition, what can happen is that this antibody can directly opsonize the microbe. So that's a good word. So it's a key word. So antibodies can act as opsonins in the process of opsonization. So what is the process of opsonization? So when a phagocyte, such as a neutrophil or a macrophage, wants to phagocytose an extracellular microbe, then basically it needs to bind to molecules on the surface of the microbe. And basically, if the microbe is covered in antibodies, it can bind to this fixed region of the antibody, basically, and that will promote the phagocytosis of this microbe. So if it finds a cell that is covered in antibody, then that is a giveaway sign that it should phagocytose it. So basically, opsonization is often compared to making the microbe more tasty, sprinkling sugar on it or something along those lines. It makes a phagocyte more likely to phagocytose that microbe if the microbe is covered in antibody. Okay, so there's another way that the antibody binding to the surface of the microbe is going to promote the death of this microbe. So it's going to activate the classical complement pathway, which is going to lead to the attack of this microbe, and it's also going to directly promote opsonization of the microbe by increasing the probability that it will be phagocytosed by a neutrophil or a macrophage. Okay, so that's the use of antibody, and it's a very powerful action against these extracellular pathogens. So that really is humoral immunity. One last thing I want to say is what these memory B cells will do. So memory B cells will go into the circulation, okay, and basically they are not secreting antibody, but they are waiting. Okay, so I'll show one of these here. And basically, all they need is the signal. All they need is to find the antibody again. And if they find the antibody, they will instantly activate. Okay? So, sorry, not the antibody, if they find the antigen. So if you get infected with this microbe again, and the antigen comes back into the blood, so here it is in blue, then it will find these memory B cells here that are in the blood, and it will end up binding to the B cell receptor on the surface of these memory B cells. And what will happen now is that this B cell, this memory B cell, will instantly be activated. So it doesn't need to go through any of that faff that we went through before involving the T helper 2 cells. Instead, it's going to be instantly activated, basically. And what will happen is this memory B cell will just divide and differentiate into a huge number of plasma cells, okay? So here's one plasma cell, but it's going to form a huge number of them, so it's going to divide and differentiate. Okay, so it's going to make loads of plasma cells, which will instantly start producing antibody against that pathogen again. And this is why, once you've had a certain infection, you're unlikely to get it again, because you have memory B cells in your circulation, which are sitting, and as soon as they see the antigen, they will just activate and produce a whole colony of plasma cells, which will start chucking out this antibody very quickly, and that will lead to the destruction of this microbe much quicker, basically. So this is the plasma cells. Okay, so that's the role of these memory B cells. Okay, so we'll continue our discussion of the adaptive immune system in the next video, where we'll move on to the cell-mediated immune response.